Hello and welcome to Lab Roots, the Life of Her Mind, an interview series devoted to learning how women think about their research, their field, and their future. Let's get started. Today, we're in conversation with Dr. Gina Turrigiano, Joseph Levitan Professor of Vision Science in the Biology Department at Brandeis University, where she heads the Turrigiano Lab. Dr. Turrigiano completed her undergraduate studies at Reed College and her PhD at the University of California, San Diego. Publishing on an array of topics in neuroscience since 1997, Dr. Turrigiano has long focused her research on brain plasticity. Recent research furthers the understanding of homeostatic plasticity mechanisms through studying sleep-wake cycles, and the results of her research include advancements in understanding how so-called self-tuning mechanisms relate to brain disorders like epilepsy and autism. Dr. Turrigiano's work has been recognized with fellowships from the MacArthur Foundation, the Genius Grant, and Sloan Foundation, and awards from the McKnight Foundation, the National Institutes of Health, and the Human Frontier Science Program Organization. A member of the American Academy of Arts and Sciences and National Academy of Sciences, a fellow of the American Association for the Advancement of Science, and the president of the Society for Neuroscience, Dr. Turrigiano is a groundbreaking researcher working at the leading edge of neuroscience. Welcome, Dr. Turrigiano, and thank you for taking time to give us a glimpse into the life of your mind.
Thanks, Mia. It's great to be here.
Okay, so fun fact, within 10 years of completing your undergraduate degree, you became a faculty member at Brandeis. That's pretty extraordinary. How did you do it?
Well, first, I would say that for my generation, this is a little bit less extraordinary. So postdocs were not as long back in my day as they have become. And, part of that is just the overall increase in sort of competitiveness of science, which is, it's papers are more complicated now. They take more approaches. And so it tends to take longer for postdocs to bring projects to fruition. So I sort of zipped through my postdoc in like 4 years, which is pretty unusual, certainly now. But back then it wasn't quite as unusual. And then the other thing I would say that's sort of interesting actually is that I probably went on the job market a little bit earlier than I should have. My main paper wasn't out yet. And my husband and I had a two-body situation, and basically we had in the end offers on opposite coasts, and it was a bit problematic because we already had a small child. So we ended up taking a position at Brandeis University together, and we started out sharing a position. So we had half a tenure track faculty position each when we started. So it was a little bit of an unusual situation in many ways.
Right. Now, can we drill down on that a little bit further, mainly because it is the case that in academic life, partners oftentimes have to make tough decisions. You mentioned having received offers on two coasts. So what was the thinking process behind settling on Brandeis, and how did, if I may ask, how did Brandeis come up with this sort of shared position that, of course, eventually split off? So
right.
Running your own lab.
Yeah, well, Brandeis had made an offer to Sasha Nelson, my husband. They were really interested in a slice electrophysiologist, and that's what he was doing. I was doing very different things at that point. Mm-hmm. And I was a postdoc at Brandeis, so I knew the place pretty well. And I had not applied for the position at Brandeis, in part because I was a postdoc here. So actually, Sasha had the great idea, well, why don't we actually go to Brandeis and just make the suggestion to them and see what they say? And it's sort of a testament to Brandeis's creativity and entrepreneurship that they looked at this crazy idea, and instead of saying, oh, no, absolutely not, they were like, maybe we could make this work. So it was extremely unusual. I don't think anyone else has ever done it. I don't think Brandeis has ever done it since. And so I would recommend taking half a tenure track position. But in our case, it worked out. I think we had faith that Brandeis was operating in good faith, that they really wanted this to turn into 2 positions. Mm-hmm. So that reassured us that this was a risky, crazy idea, but maybe it would work. And it turned out to be a great thing for us, a great thing for Brandeis. Yeah. So. Yeah.
Well, what's one of the things that I think is so interesting about the story so yes, it's unique, it's unusual. As you mentioned, Brandeis probably hasn't duplicated the or hasn't made a practice of it. But what's interesting, certainly for people involved in relationships coming out of out of grad school or in a postdoc, knowing that they can be creative, that they can at least ask, as you said, Sasha said, ask the question. Why not ask? The worst they can do is say no, right?
Yeah, and I think what was really critical about this was certainly at that time in history, a lot of places still wouldn't hire couples. I mean, not every place, but mm-hmm. It was changing. But there were still places where that was considered problematic to have 2 people in the same department who were married. Mm-hmm. And so I think the sort of flexibility around hiring was really much things were not as flexible at that point in terms of hiring couples. It's become, I wouldn't say every place accepts that this is, if they really want to recruit good people, it's something they have to take into account. But it's certainly much more common now. Mm-hmm. So it was a creative solution. I mean, I think the best solution, of course, is that places come up with an extra position, not half a position. Oh, but what I wanted to say was the critical thing about how we structured it was a lot of places would, at that point in time, would make an offer to usually the man, and the woman would have an adjunct position that wasn't on the tenure track, with the idea that maybe they could come up with a tenure track position down the road. And that's not the way this was structured. It was structured as we each had half of mm-hmm. the FTE, right? So that meant we were on equal footing, and it meant that, it was sort of an absurd situation that couldn't last for very long, but it meant we kind of came in without an imbalance to begin with. So there were things about it that were really very creative and very kind of an attempt to be, sort of progressive about it. And yet, it was also problematic because we were sharing one lab space, one salary. We also we're sharing our teaching responsibilities, which had its upsides and downsides. So, it was an interesting couple of years for starting out the lab.
So it lasted about a couple of years before they split
I think, I'd have to go back and check exactly. It lasted about 1.5 years. And at that point, I got a K99. No, sorry, not a K99, a KO, one of the K grants. I forget what it was. That basically paid my full salary.
Oh, nice.
And that was enough for the chair of our department to go to the university and say, hey, look, it's not going to cost you anything until after they would get tenure anyway. So why don't we just make it 2 positions? And the university converted it. So it wasn't very long.
Yeah.
So it ended up being a great overall creative mm-hmm. approach that resulted in both of us being at the same place with the tenure track job, but it wasn't so clear it was going to work out at the beginning.
But it was worth the risk in any case, and certainly in retrospect, definitely worth the risk because of how it turned out. Yeah, well, congratulations in any case on coming up with this really cool solution and working with an institution that was open-minded enough to say, hey, this let's try it. Why not? It can work. Okay, so now I want to back away from the sort of the job, establishing the job, to ask this question. So why science? And maybe I'm not thinking about your undergraduate institution the right way, but when I think of Reed College, gorgeous institution, really high caliber intellectual activity, but I don't think specifically of neuroscience. I think more of just broad-based liberal arts education. So So why science?
Right, right. Well, I think I'm one of those unusual kids that declared at age 9 that I was going to be a biologist, and then I actually became one.
Yeah.
So,
Okay.
but at that point in my life, I thought being a biologist was going to be, being some kind of population biologist or ecologist out doing field work, and working with animal populations. That was kind of my dream. So, Reed was an amazing place. And I think a lot of people who are interested in science, or especially neuroscience, have a very broad set of interests, right? I mean, we get into neuroscience because we're fascinated by the brain, right? The brain is what makes us human, lets us learn, understand, try to comprehend the universe. It sort of intersects all of these other things that, philosophy, literature, I mean, all of this stuff kind of comes into it when you're thinking about how the brain works and what it does. So in some ways, it makes a lot of sense. I think that somebody interested in lots of things, but primarily in science, ends up working in neuroscience. It just speaks to so many elements of human existence, right? So I kind of went in to Reed knowing I wanted to study biology, and it was a great place to be an undergraduate in science, in part because you still were steeped in all of the liberal arts stuff, which I loved.
Yeah.
And then the other piece of it is that everyone there does a thesis. So everybody does a full year of research. And, if you're in the sciences, it means you're working in a lab and you're actually writing a senior thesis. That's a research, you're writing up a research project. So it's pretty amazing preparation for graduate school. So that's, I would say neuroscience, I wasn't at all thinking about neuroscience until I took my first neuroscience class. And then it was sort of like, being struck by lightning. It was like, oh my God, this is the thing that I want to do.
And just so a complete shift from thinking about broadly construed environmental concerns, non-human biology, to, whoa, brain is it.
Yeah, not of course the human brain, because I study rat and mouse brains, but nonetheless, yeah.
And so is it the case then that neurobiology, so for someone, because the neuroscience is this big field, yes, it's oriented around, all things brain, but it's this really enormous field in terms of how you can go about specializing and subspecializing. How did you come to then neurobiology? So you were interested in biology, you took the neuroscience class, and you just went, aha, this is it. Like, how do you remember how that struck you? Like what the details were that you were looking at?
Yeah, pretty much. I think it just seemed incredibly fascinating. And then the next step was that I started to do summer research. So a friend of mine had been working at in a neuroscience lab in Portland. And so I ended up getting a job in that same lab over the next summer. And then that sort of set me up for doing my senior research. So, and then it was just pretty obvious to me as soon as I started working in a neuroscience lab that was what I wanted to do. Yeah.
And then there's brain plasticity. So it's a term that, we hear on the news, or we might, read somewhere, but, most of us who don't study the brain from the standpoint of scientific investigation, we think plasticity. Wait, does that mean that, the brain moves around, or that, if you're missing a part of the brain, another part of the brain can fill in? In other words, like, how should someone like me think about the concept of brain plasticity?
Right. Well, it's a really big umbrella term, and there's a lot of different things that it encompasses. You could think about changes in the brain when brains develop. So, if you've ever watched a little kid figuring out, I don't know if you have children, but, I became quite fascinated in development as soon as I had kids, because it was amazing, like watching the things that they couldn't do one day and could do the next day through trial and error and just brain maturation. So that's a form of plasticity, meaning the brain is changing during development, and basically the brain is interacting with the outside world and getting feedback, and that feedback is being used to make brain circuits better at what they're trying to do. So the circuits that control reaching, the first time your kid tries to reach and grasp something, it's not going to be able to do it, and it takes a lot of practice and changes in the brain to make the kid able to do it. So all of those changes are basically affecting synaptic connectivity within their brains and making those brain circuits able to accomplish the job. So it's like this incredible process of tuning these circuits up and matching them to the environment. So that's one form of plasticity that I personally am very fascinated with and that we study a lot. And obviously this has implications for all sorts of disorders where that neurodevelopment doesn't go according to plan, right, for whatever reason. And it also raises interesting questions about what kind of experience we need as a young mammal to actually make our brains work properly.
So am I thinking about circuitry and synaptic connections the right way when I talk about my mental experience of, let's say, trying to think through an argument, right? So, I've got, let's say, a chain of reasons or long division, that sort of argument, right? And I've got to get from one idea to the next, to the next, to the next. And suppose that I get stuck, like I don't know how to, divide 32 by 4, something like that. So I'm stuck. Am I right to think that there's this electrical firing of some sort of chemical that's kind of reaching out, but not quite grabbing the next neuron? I mean, I'm sure I'm messing up a whole lot of language here, but that's like one way that I think about it. And then you were mentioning how so forget just thinking, but when we're interacting, when a child, let's say, is learning how to reach for and successfully grab something on the table as they're working out depth perception, is it also something like what I've described, where when you're talking about circuitry and synaptic connections, is it the brain's trying to connect up parts of itself with other parts?
Yeah, pretty much. I mean, look, you've got a whole bunch of neurons in your brain, 100 billion or something, give or take. No one's exactly sure what the number is. And all of those neurons are connected to other neurons. And how many connections are there? Well, some neurons have maybe only a few hundred. Some of them have hundreds of thousands of connections. So you have this unbelievably complicated interconnected network. And it's divided into subnetworks that control different aspects of our behavior. But each of those networks, that wiring is very complicated, obviously. Somehow you have to fine-tune that connectivity to enable all of these complex behaviors that we can do. And that's true. It's easier to think about in terms of a motor movement. But similar things have to be true when we think about our ability to reason or to sing a song or, parse a complicated philosophical argument, right? It's using different parts of our brain, but it's the same sort of principles. That said, we don't exactly understand this process, right? That's why neuroscientists like me are still in business.
Yeah, and you will be probably for some time.
For long, certainly long past when I'm actively doing science.
Well, so then, okay, so you've mentioned in talking about, so when you talk about singing a song, I think, memory. You talk about, reasoning our way through an argument or sort of unpacking a complicated concept. You are probably using memory, but you're also using some other feature or features of the brain. And then you also mention disorders. So, for example, you've published work on that's related to epilepsy and autism. I don't know which, if any, you can help us understand in terms of this idea of circuitry. And then you've got this really interesting concept of fine tuning, which, if I understand correctly, you think of or you work on in terms of this sort of internal system. It's effectively a closed system of fine tuning. Is that right?
Well, not necessarily, actually. I think one way to view it is that so the other piece of this that we're really interested in is how it is that you can that these networks, these highly interconnected networks that are very plastic, where these connections can change all the time. Mm-hmm. How is it that they maintain some sort of stable function over time? Mm-hmm. So, you have a sense of self that is very constant, and pretty much you can sort of trace it back all the way to when you were a little kid. You've had this continuous sense of self and things you're interested in. And so despite all of the change that you've undergone, there's this sort of core stability to essential things in your brain, essential brain functions. Mm-hmm. That's one way of thinking about this at a very high level. When you think about sort of the nitty-gritty of the actual circuit, it's really hard to do that. It's actually hard to take a circuit that is really plastic, where all the connections can change all the time, which you would want to learn, a new motor task or something, without that circuit sort of becoming dysfunctional. And the reason is that these circuits are composed of excitatory and inhibitory elements, and they're highly interconnected. So you could think about a feedback amplifier. Like imagine Jimi Hendrix playing his guitar with the feedback, right? Okay, that's this positive feedback loop where you're getting, that sort of ramps up and the sound, and it gets out of control, right? If it gets too intense. Well, our brain circuits work the same way. They're these little amplifier circuits. So you have some neurons that are excitatory connected to other neurons that are excitatory, and they feed excitation back. So you have these feedback loops that can get completely out of control, right? So the way to fix that is to have inhibition. So some neurons can actually turn down the excitability of other neurons. And what that means is in these circuits, you have to balance the excitation and the inhibition pretty carefully to make sure you don't end up with this runaway feedback. Does that make sense?
It does. And a number of ideas come to mind, but given your work on the relation between the amplifiers and the modulators or inhibitors, is it the case that you're saying something like this? If so let's say we have someone with a psychiatric condition, depression, anxiety, something that's fairly common among a lot of people, or something less common, but still, fairly prevalent, like epilepsy or autism. Are we saying that part of the search is for bringing to bear or introducing one of those sort of modulating or inhibiting circuits into an existing sort of feedback loop where there's this sort of spiraling out?
Yeah, that's kind of, this is one idea for some of the things that could go wrong in some neurological disorders.
Okay.
The idea is that normally you've got this potential runaway excitation, right? And then you've got inhibition that can come in and keep it in check. And the question is, how do you get that balanced right? Because if the circuit is not excitable enough, if you can't amplify signals, then your brain doesn't work properly. You can't detect sensory input. You can't process it properly. If that excitation is too high, then you have the opposite problem, right? You end up with this sort of runaway excitation, which puts these circuits into a, kind of an epileptic state, this hyperactive state. And then it doesn't function properly either.
Mm-hmm.
So what this says, so this brings in this concept of homeostasis, which is what we've worked on a lot. And that's basically the idea that you can build a self-tuning mechanism that keeps that balance properly set. So if excitation goes too high, it'll detect that and bring in more inhibition to compensate, and vice versa. And there's a lot of ways you could do that tuning, but that's the basic concept. It's sort of very key, actually central concept in physiology, this idea that certain things like our body temperature, blood glucose, all these things are under very tight negative feedback control. So if they deviate from some set point, 98.6, pretty close, right? If our temperature deviates, all these mechanisms kick in to bring it back. Mm-hmm. So the idea is that circuits in our brain work the same way, that there are plastic rules that essentially sense, what state the brain is in, and then tune it back to that sort of set point where it's working properly. And so the corollary of that is that in some disorders, possibly that homeostatic process is not working properly. And so the circuits end up mistuned in some way. Now, how big an effect is this? How much of some of these disorders does this account for it? We really don't know the answer to that. What we know is that using some, for instance, genes that are very high confidence genes that cause autism in humans, if you take those genes and you mutate them or get rid of them in mice or rats, then you see behaviors that are sort of congruent with the idea that, you've recapitulated some phenotypes in the rat or mouse model. Mm-hmm. And what we see is there are defects in these homeostatic mechanisms. And so, what's the causal relationship? That's complicated. It's going to take a lot of work to get at, but it does raise a sort of interesting avenue for trying to figure out how you might try to reinstate these homeostatic mechanisms, and would that have therapeutic value? So that's certainly one of the things the lab is interested in. It's a long way to go from work in rats and mice to really finding things that are effective in humans.
That so when you were talking about the analog in the rest of our physical systems, right? That I was thinking, well, yeah, of course, that makes perfectly good sense. But it wasn't until you articulated it, as you did, that it sort of crystallized. And what jumped out for me was I was thinking of I developed a Hashimoto's thyroiditis when I was quite young, and it struck me over the years that so the brain is screaming out for more of a hormone that my body isn't producing, and I often wondered, so okay, endocrinologist, why aren't you all sending me to some sort of neuroscientist, like a neurochemist or a neurobiologist, somebody who can try to help me understand what's happening in the brain, but also what the corollary is for other ways that my body is feeling reflected in the brain. So I was thinking that, but then I was wondering, so given some of the focus of your research, I was wondering kind of what other analogies do you use as a way to think about what's going on in the brain, since you're basically mapping the territory that hasn't really been mapped before, right? So this is all new, and you're having to find ways to conceptualize what it is that you're doing. Am I right to think that one reason why you focus on sleep-wake cycles is because you're trying to use sleep and wake as, I don't know if analogy is the right term, but as a way of getting at this homeostasis that you mentioned before, the homeostatic mechanisms?
Yeah, I would say our work on sleep and wake sort of kind of came. We started it for a different reason. I mean, part of it was because let's see. Part of it was based on some assumptions that turned out to be kind of wrong. Like we basically had this idea that, well, okay, if circuits of the brain have a kind of activity set point, right, and so they're homeostatically regulating around that. If excitation goes too high, turn down the gain, and vice versa. Well, sleep and wake are such dramatically different brain states that we thought maybe it would be impossible for a neuron or a circuit to have a set point that would stabilize it across those different states. So we thought, possibly these mechanisms could be turned on or off during sleep or wake, or maybe the set point of a neuron might actually shift during sleep or wake in some way. So the circuit might be set. It might have a modifiable set point that would change as a function of brain state. So it turned out actually when we did these sort of chronic recordings in rats, where we basically let them run around for days, we just recorded the activity in their brain when they were doing just their normal stuff, but sleeping, waking, eating, playing. Now, it turned out that for sort of these normal sleep and wake states, the overall level of activity, the average activity of the circuits are actually pretty similar. So that the original motivation turned out to be wrong, but at the same time, we discovered this sort of fascinating thing, which was that the direction of compensation depends upon the sleep and wake state. So it turns out that, so you can think about a system, right, where the activity has fallen below the set point or activity has gone above the set point. So it's sort of a bidirectional system, right? Homeostatic mechanisms have to be bidirectional, or they don't really give you a stable output, right? And what we found is that the upward compensation only happens when animals are actively awake and sort of running around in the environment and doing stuff, whereas the downward compensation only happens when they're asleep. And this was really kind of crazy because at the molecular level, there's a fair overlap between the mechanisms that drive upward and downward. So we had thought of it as a single switch, like a thermostat, single thermostat switch. And it kind of suggested that's not right. There's some sort of higher level control over the mechanism. So, so it's really quite fascinating. And I would say it suggests that one of the functions of sleep and wake states is actually to segregate different forms of plasticity to make sure they're not happening at the same time. And exactly why that would be is a big, really fascinating research question that we don't really have a great answer for at the moment.
So as you're talking about this most recent finding, most recently you're talking about this finding, I'm thinking a couple of things. One is the way that you articulate what's very complicated, certainly for a layperson, right? I don't have the vocabulary. I don't have the sort of the technical concepts to help me make sense of what you're saying. But there's a level of understanding that you're helping me achieve because of the way that you're formulating the words you're choosing, the metaphors or analogies that you're using. And it makes me wonder how you go about either initially forming a hypothesis, as you did with the concept related to sleep and wake cycles, but then also how you go about either rethinking altogether or recalibrating, as you did when you said, oh, what we thought was happening isn't quite what's happening. So how do you go about thinking through this stuff?
Right. Yeah. I mean, I think that's one of the most fun things about science is when, it's fun when you have a hypothesis and you go in and test it and it's right. There have been a few of those amazing moments where you're like, oh, it really works that way. But it's also, if you could figure it all out just by sitting at your desk, right? Why do the experiment? So it's even more in some ways exciting when you go in with an idea and it turns out you're just wrong, and you have to look really carefully at the data and think about, what does this mean? Okay, so of course you have to worry about, did I do the experiment right? All the right controls, all of that. That's the boring reasons why things you might have got things wrong. But once you've ruled that out, to me, it's sort of, I love that moment where you realize you're standing in a, you're looking into a new environment, and you've never seen it before, and you don't know how it works. And it's like, I don't know, it's like being an explorer on the first one to see some exciting terrain. It's kind of an amazing feeling. So it's also scary, right? And often for the grad student or postdoc, because of course I'm not doing the experiments myself anymore. I've got this amazing crew of people, creative, wonderful people who are doing the experiments, and it can especially be difficult when their cherished idea turns out to be wrong. But I do think these are the moments where we learn the most, right? Mm-hmm. So it does. I think my research tends to be pretty idea driven. I mean, we do like to just go and look and see what we see, but we're usually going in with some roadmap that arises out of some theoretical construct for how we think it'll work. And that's really useful. Like, it helps you design your experiments properly. It helps you come up with the right experiment when you're trying to really differentiate between these two possibilities, right? But it's also very easy to get sort of fall in love with your ideas and cherish them too much, and it can be very hard to abandon what is a very beautiful idea when it turns out that idea doesn't really describe what you're seeing. So it does take a certain fortitude to be able to remain flexible in that way. But that's sort of the hallmark of, that's when science makes progress, right?
Am I overstepping when I say it's basically the concept of plasticity writ large in the discipline, where you try something out, and if it works, great, you can stick with it. And then if it doesn't work, well, now you've got to try to make a new connection.
I think that's absolutely right. And that's what, often we're stuck as scientists. We have these pieces of data that it's pretty clear that our theory about how it all fit together is wrong, but we don't know. I mean, we don't have enough information to know how it goes next. And so, how do you keep your mind open to these different possibilities? It can be a really tenuous state to be in while you're figuring out the right next steps and where to go next. But so I do think there's a lot of it requires a lot of flexibility and plasticity.
What do you do to, so what's your go-to? You mentioned that, your work is presently very idea driven. Like, what are your go-to resources? What are your go-to tools to think about where you might go with an idea that you've been sort of percolating? Kind of how does that work? For where do you do it? Like how does all of this come together for you?
Right. Yeah, I think this is a fascinating question. That generalizes across. I mean, this is just a question about human creativity. Where does it come from?
Yeah.
And
answer
whether it's in the sciences or anything we do, right? I think science is interesting because at the same time you have to know a lot, and you have to do a lot of work to get to the point where you can have a good idea, right? So you have to read the literature, you have to know what people have done, you have to really understand things at a very nitty-gritty level. So you have all this detail, that you have to somehow take into your brain, but that's just not enough, right? Mm-hmm. You can't then sit there and sort of logically hammer out, well, therefore it must be this way. At least that doesn't work for me. Mm-hmm. So, you bring in all this stuff into your brain and then it kind of rattles around in there for a while. And then something happens. And, I can't explain what that is. I've had actually graduate students, when I go to visit universities, you have lunch with graduate students and postdocs. And it's really a wonderful practice. They get to ask you all sorts of questions. But a lot of times they'll ask, where do your ideas come from? And, how did you have this idea or this idea? And I can't tell them. I mean, obviously, most ideas have all sorts of precedents in the literature. People have thought of similar ideas, So of course, what your brain is doing is taking these pieces that you brought in and it's rearranging them and coming up with something that is based upon all of this other stuff, but it's a new variant, right? And I don't know how that works. I mean, the only thing I can say is that it works best for me when I'm not in the lab. So it's usually when I'm out, out on a hike or something like that, right? When my brain is kind of free floating is when I have most of my ideas. So I tell this to people in my lab, look, you got to work hard, you got to do all these things, but you need the downtime because that's really when a lot of the creative stuff happens, at least for me. I think everyone's probably really different that way, but.
Right. I mean, some people might do just what you said doesn't work for you, namely sort of, really hammer out, as you say, sort of the implications of a set of ideas. And then some people will do, as you say, you do, which is disconnect yourself from the lab and from sort of doing science, and you're just being a human in the world, engaged in various activities that somehow maybe give you time to digest or percolate, but also maybe give you new ways of thinking about the stuff that's percolating. And I think it's really helpful for people to know that they have to find what is going to work for their sort of temperament or their sort of intellectual disposition or whatever it is that gets them, that sort of characterizes them being in the world. So thank you for sharing that. I've got two final questions. One is, given how young neuroscience is as a discipline in its own right, kind of how do you keep up with stuff going on around the world that's relevant to what your work is, on the assumption that that's happening? And then secondly, what do you want to see happen like within the next 10 years?
Right. Well, to the first, actually, COVID was a really interesting experience to go through because most of us were scientists, and I mean people in general, but we were sort of cut off from our usual modes of interacting, which is we travel places, we meet other scientists face to face, we have conversations about what we're doing, they tell us about their work, we tell them about our work. There's this kind of talk about sparking creativity. It's like those personal interactions somehow are just way better than what we were getting over Zoom. So that's the way we usually do it, right? And actually, the last couple of months, I've been traveling again, and of course, being mindful of carbon footprint and all that. There's great things about, so it's complicated, but obviously we can read the literature and we can do all of those things. We have to do them to keep up with stuff, but there are, thousands and thousands of papers. I mean, there's something like, 30,000 papers a year on plasticity. I don't know. It's, I made that number up, but there's a lot. Right? So we just can't keep up with all of that. So you have to be selective somehow. And then it turns out that that's just not a very, humans are just not good at absorbing information that way. I mean, it can be very efficient sometimes, but in terms of like paying attention to things and having it spark new ideas, it's really quite a bit better to be interacting with people live, So scientific meetings are really an important way that scientists interact with each other and get new ideas. You're learning about stuff that's happening long before it ever gets published. So of course it's much more on the cutting edge, but it's also something about the face-to-face interactions that I find at least really make it much more effective. So that's a real dilemma, I think, as we're going forward. We're thinking about accessibility. We're thinking about being responsible citizens. It's hard for us, I think, like for many people to figure out how to balance those. And then I forgot what your second question was.
If you could, so I'll reformulate it slightly. If you could have one question answered within the next 10 years in your field, what would it be?
Oh my God.
I know, it's horrible.
What is consciousness? No. Not really. That's too hard. That's just too hard. I guess I feel so. One reason it's hard is that I don't think I think the way we're going to understand the brain is slowly and sort of holistically. Like we're not going to understand one piece of it and then not the rest of it, because the problems are all the same problems, right?
Okay.
So I think the field is going to keep progressing, but in a way that is, we're really groping towards trying to understand something as complicated as the brain is. And so, I guess if there was one thing let's say I would love to have figured out maybe by the time, by the time I retire, I guess it would be, it'd be really cool to understand, say, what the mechanism is by which sleep can switch homeostatic plasticity on and off and control the direction. That would be really pretty cool to understand. And I would really love to understand how these processes that we've been studying for a long time, how they actually enable our brains to store lots of information. So, we think these homeostatic mechanisms probably increase the capacity of networks for memory storage. There's sort of good theoretical reasons to think that based on a lot of other people's theory work. So it'd be great if we could figure out why that is. So those are sort of two threads that are going on in the lab now that are pretty exciting. But overall, these are like tiny, they're sort of big, but they're also sort of tiny problems. And I think that's where everyone's at right now in brain research. Like we hopefully are going to start to get a handle on some of these very fundamental processes. And once we do that, then I think we can start to tackle some of the bigger questions.
Oh, lovely. Well, I hope that within the next 10 years, I'll be reading about your answers to both of those questions, and we'll be able to, as you say, move on to larger and larger questions. But in the meantime, thank you so much. I appreciate your time and expertise. And I know everyone is going to be feel very lucky and be very lucky to have heard from you directly. So thank you, Dr. Gina Turrigiano, for your time. And I hope that we will get to talk to you again someday.